Video is just a quick overview of the nervous system. Okay, so the organization of the nervous system. We know that there's the central nervous system and the peripheral nervous system. Central nervous system is the brain, which is protected by the skull, and the spinal cord, which is protected by the vertebrae. Your peripheral nervous system, you've got 12 pairs of cranial nerves. Some of them are afferent, some of them are efferent, some of them are mixed. Spinal nerves, there's 31 pairs. They're all mixed, and so there are sensory and motor divisions of each, afferent and efferent. The autonomic nervous system is also known as the visceral nervous system, and so this is all involuntary. Oh, I guess I should mention motor for your spinal nerves, the efferent motor divisions of those nerves are involved with voluntary movement. So, like if you're doing a bicep curl or some kind of walking around, you're voluntarily innervating those muscles using the motor division of your spinal nerves. By contrast, autonomic nervous system, this is all involuntary. So, signals to your digestive organs, smooth muscle lining blood vessels, the walls of your gastrointestinal tract, and the cardiac muscle of your heart is supplied by the autonomic nervous system, and also glands. So the autonomic nervous system can further be divided into sympathetic, which is generally going to be speeding up processes, and parasympathetic, which is going to slow them down. So your vagus nerve innervating your heart, or your phrenic nerve to the diaphragm are good examples of autonomic innervation. Okay, neurohistology. So what kind of cells make up the your nervous system? Well, there are nerve cells, aka neurons, and there are glial cells, aka neuroglia. So first we're going to look at our neurons. Okay, so what are the functions of neurons? They have obvious sensory and motor functions. By conducting impulses, they're able to store and retrieve information. So neurons, structurally, they have a cell body, or the soma, and there are going to be two types of processes coming off of them. Axons, which convey impulses away from the soma, and you're only going to have one axon per neuron in vertebrates. And then dendrites, there can be one or there can be many, and so these are conducting impulses towards the soma. Let's just take a look at what we have here. So we've got the soma, we've got many dendrites coming in, we've got one axon. We want to see this myelin, which is the function of that is to speed up the transmission of neural impulses. We want to note this Nissl substance, and all that is rough endoplasmic reticulum, and it's found everywhere except for at the axon hillock, where the axon originates. So myelin, as we were discussing, it's a lipoprotein sheath. And we're going to get into some more things that differentiate, or differences between central and peripheral nervous system with respect to the myelin. We want to see the initial segment here of the axon, which has the lowest threshold for excitability. Okay, so axons can vary in length. Very short axons, they can be very tiny. As an example, the granule cells of the cerebellum, the granule cells of the dentate gyrus of the hippocampus. These are both places where it's possible to get a cell division of neurons in adults, which was once thought to be not possible. Also, the olfactory epithelial cells will have these small axons. Well, the epithelial cells don't obviously are not neurons, but like embedded in them are these neurons that have very short axons. Okay, so yeah, these guys have rapid turnover. An example of a very long axon would be an anterior horn cell and spinal cords. When you move your big toe, there's neurons, axons that are several feet long that are allowing that to happen. Okay, so in an axon, it's important to know that there are neurotubules and neurofilaments, and these provide structure to the axon and also direct axoplasmic flow. As promised, myelin in the peripheral nervous system is made of Schwann cells, and you can have many Schwann cells myelinating a single axon. In the central nervous system, the myelin is formed by oligodendrocytes, and you can have one oligodendrocyte that's myelinating many axons. Demyelinating diseases are absolutely bad. Multiple sclerosis, for example. And if you have demyelination of the phrenic nerve, that would be really bad because your diaphragm is going to be getting less innervation, and if your diaphragm isn't working, you can't really breathe. So that's bad. Okay, so on our axon, we've got a Schwann cell. This is one Schwann cell, the nucleus. This is another one. In between them, there are spaces that are not myelinated that are called nodes of Ranvier. So in saltatory impulse transmission, you've basically got like jumping between the nodes of Ranvier. So the Schwann cells in that way speed up the transmission. Okay, on to dendrites. You could have one, or you could have thousands. Basically, these guys have like these little spines on them that increase their surface area, and one dendritic spine can have, as many as 9 axons synapsing with it. So that's all we want to see there. So those are neurons. Moving on to glial cells. In general, these are the metabolic and support cells for neurons. So these guys help neurons do their job. You can divide them into macroglia and microglia. 2 examples of macroglia are astrocytes and oligodendrocytes, oligodendroglia. What do we want to say about these guys? Okay, so as an aside, like in embryonic development, all tissues are derived from these 3 germ layers: ectoderm, endoderm, and mesoderm. Ectoderm, so your neurons and macroglia are derived from the ectoderm, and microglia are derived from the mesoderm. So how do the microglia get to the CNS? They migrate from the bone marrow along blood vessels to the central nervous system. Very good. Astrocytes. This is my ghetto drawing of an astrocyte. So they have many processes coming off of them that are all alike, and there are many functions to the astrocyte. They initiate the formation of the blood-brain barrier. The blood-brain barrier is the actual tight junctions between endothelial cells in the brain capillaries, and right, it's like these foot processes of the astrocyte are like forming or like abutting those tight junctions, and that's sort of like the gatekeeper between like what's going on in your blood vessels and what's going on in your actual brain. Okay, so other functions of astrocytes: they can mop up excess potassium that's left in synapse after neuronal firing. They help form the glial membrane. So in this little drawing here, you've got the brain, and it's covered. The closest layer to it is the pia mater, and so you've got these support cells, astrocytes in your brain, and the foot processes are budding, joining right up against that. And so the function of this glial membrane is to surround and protect the entire nervous system, and the astrocytes are an important part of that structurally. Just note that most of the time a brain tumor is not a tumor of neurons, it's a tumor of glial cells. So most brain tumors are glioma. Continuing with the function, this is the last function for astrocytes. They provide a scaffolding for neural migration and development. Okay, oligodendrocytes, which was the other macroglia that we looked at, these form myelin in the central nervous system. and they serve as satellite cells to blood vessels, neurons, etc. Satellite cells, just places where, you know, important substrates can be stored and then delivered to these cells. Okay, microglia. What are the functions? Well, generally microglia are very small until activated by an injury, and then they become phagocytic. For example, in Alzheimer's disease, you're going to see a lot of active microglia. There are many types of microglia. That's just about all we're going to say there. Okay, let's get on to the synapse. So this is the structural and functional unit of the nervous system. This is really where the action takes place. So in my little diagram, this is going to be an axon. This is going to be a dendrite. Generally, the synapse is this area. The synaptic cleft refers to the actual gap between the axon and dendrite. So as a general, sort of very simple schematic, you've got an electrical impulse traveling down the axon to the presynaptic terminal. This is where there are vesicles of neurotransmitter are formed and stored, sort of waiting for action. And what happens is, as this electrical signal reaches the presynaptic terminal, there's going to be an influx of calcium ions, which is going to set up the, quote, substructure, which is going to guide these vesicles to the presynaptic membrane. And just to continue there, when that happens, the vesicles will fuse. You'll have a release of neurotransmitter into the synaptic cleft. An example of a neurotransmitter, and there are many, many of them, but glutamate is the most sort of prevalent neurotransmitter in our brains. So these neurotransmitters are then binding to postsynaptic receptors, which is going to cause ion channels to open, and that's going to basically set up the electrical signal being transmitted down the dendrite for further action, so to speak. So the big picture is you've got an electrical signal, it sort of gets converted into a chemical signal at the synapse, and then it gets reconverted back into an electrical signal to travel down the dendrite. So what happens to neurotransmitters after they've been released? So they can be broken down or hydrolyzed, at which point they're no longer going to be activating the postsynaptic receptors. Or they could be recycled by hopping on some sort of carrier back into the presynaptic terminal. So an example of this system being sort of abused is when you take cocaine. What happens is the cocaine molecules hijack these carriers. And so those carriers are no longer able to accept dopamine, which is what they're supposed to be carrying back to the presynaptic terminal. And so the result is you've got all this excess dopamine in the synapse. And that's what makes you get high until it wears off. Okay, so one important note here is that neurons that fire together wire together. And the sort of basis for that is when you've got signals crossing the synapse, in addition to an electrical signal being transmitted and carrying down the next neuron. You've also got the potential for the activation of genes within the postsynaptic neuron, and these genes, once activated, will lead to the translation of proteins, which can actually sort of beef up the structure of this postsynaptic dendrite. And so the idea is that, like, the more active a given pairing of axon and dendrite becomes, like, literally the bigger and more, sensitive and able to pass on signals it becomes. So that's just the idea that neurons that fire together wire together. Okay, thank you very much, everybody.